Welcome to Let's Meet the Virologist, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with students, postdocs, and other virologists so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. On June 22nd, 2022, we talked with Aurelien Chuard, a postdoctoral researcher at Boston College. He received his master's from Bordeaux University and PhD from Tours University in France. He is currently studying insulin-like peptides encoded by large double-stranded DNA viruses in the Iridoviridae family. Thanks for talking with us today. Why don't you tell us a little bit about yourself?
Sure. Nice to meet you, Teresa. So my name is Aurelien Chuard. It's a very French name, quite hard to pronounce for some Americans, but still. So I'm based in Boston now for a year. I'm a postdoctoral researcher at Boston College in the Altindis lab, and I'm really happy to be with you today.
Great. And can you tell us a little bit about sort of how you first became interested in science and then virology?
Yeah, sure. Every time this question is coming, I'm always saying that it's thanks to great teachers first, because teachers can be great, some can be bad, but when you have great ones, it can give you the vibes to do science and to go on for the next year, next decade, or maybe even for life. So that's my first interest in science, and specifically in microbiology first was thanks to great teachers.
And then can you tell us a little bit about sort of the path that you followed to get where you are now? So how did you sort of end up in your undergrad or your graduate institution, this kind of a thing? What brought you to those institutions, those particular labs, and then the one that you are in right now?
Yeah, sure. As I say, I have great teachers in high school, and I was already taking some lessons, biology, specialized biology, in microbiology specifically. And so just after high school in French, we have the different paths to choose. I choose first to go on a technical degree on biology. Of course, it was more industrial, a bit medical, and so I follow this pathway to just get my first degree as soon as possible, and basically just after that maybe go to work directly. So it was 2 years after high school graduation, but after that I learned more and more about the theory of biology, and I was I want to learn more and more. So finally, after this technical degree, I went to college. So it was 3 years of college in the University of Bordeaux. The first technical degree was in Lyon, my hometown, which is really kind of famous about infectiology. We have the only biosafety for laboratory in public laboratory in France. So it takes there is a lot of resources in this town. But I moved from my college to Bordeaux, which is a city that is most famous for the wine, but still there is good research. And so during this degree, it was my it was kind of bachelor degree in France. I was more on biology, cell biology, and molecular biology. And finally, to go back on what I was really fond of during my master degree is I went back to microbiology with an opening on immunology as well for 2 years, same in the University of Bordeaux. And as in France, it's kind of mandatory to have a master degree to go in PhD programs. So after that, I applied to different PhD programs, and you have to pass some contest against other of course applicants. And I finally went to the Tours University. It's nearby Paris, 1 hour train with high-speed train. And my PhD was on virology. My master thesis was as well on virology. So I start with doing my master degree on adenovirus, so a double-stranded DNA virus. And I follow up with my PhD that was working on a Marek disease virus. It's not very famous, but really interesting herpes virus. And I did my PhD in 3 year in tour. And yeah, it was like really, really interesting because the virus that I was studying in my master, like adenovirus, and the one like the next one, the Marek, are really interesting because they are like, and all of the viruses have the abilities to like encode peptide protein that modify and modulate the environment, or like the cell environment or the cell metabolism. And my two labs was really like trying to characterize this really like interesting peptide that really can switch everything, can like have like either countermeasures or like can just perform chemoattraction. So it's really like my background now, and I will probably explain that later. Here in Altendis lab, it's exactly the same. So I always following this path of like almost viral mimicry and like also like protein that can like regulate and modulate the cells directly. So yeah, that's my path to go where I am now.
And how did you end up in Boston? What was that process like?
Yeah, sure. About Boston, like 2 years ago, like during my PhD, a really good friend of mine like sent me a YouTube video, which is really interesting. And it was this video was about the discovery of the viral insulin by Dr. Altindis in Ronald Kahn lab in the Harvard Med School. And I was really like mesmerized, like, wow, like why viruses can like have this insulin and why. So of course they start to work on this, and Emra, my boss, like built his own lab, and like 2 years after, he just like of course sent an application, a job offer, and on Twitter, and I just applied to it, and yeah, so because I was like 2 years before I see this video, and like it was just a good timing for me just to apply, and of course like we have like interview and everything. And after I just joined his lab because the virus like that encodes the viral insulin are double-stranded DNA virus. So I was working on some of these viruses. So I was a good match. So I just jumped at the end of my PhD. Actually, 2 weeks after my PhD defense, I just jumped to Boston. So it was really, really, really cool.
Cool. And can you tell us a little bit about that work, I guess? So sort of like, what is the big picture question that you're trying to address? And then maybe some of the techniques that you use to sort of do your work.
Sure. So as I say, my boss discovered like 4 years ago the viral insulin. So there is some viruses that encode in their genome. They are all double-stranded DNA viruses, so quite large virus here. And they have in their sequence some insulin-like peptides. And what he did is to chemically synthesize this insulin and try to see if there is any interaction with the insulin receptor or the IGF-1 like receptor. And apparently it works. It works really well. So basically this insulin can bind to the human receptors, and that was like a really like the founding of the lab. So virus encode insulin, and this insulin, when they are like synthesized, they can bind to the receptor and of course elicit all the downstream signaling in the cells. So that was like the very beginning, but as I say, it was like chemically synthesized insulin. So my part as a virologist, I'm like as the only virologist in my team now. It's really a challenge. So is to really understand taking the virus, like all the viruses that we have in the lab, and to understand what is the real function of this viral insulin in the viral cycle, and not only on the receptor. We have like good, really talented biochemists in the team, and they are like doing a great job on this aspect. But on the viral aspect, this is what I'm hired, and that's the goal. So that the big picture is to really understand the function of the insulins, of this viral insulin. So, and after about the technique, I really start like almost like the project or the virology project here. So it was really basic virology, but I have to really start from almost nothing. When I jump here, it was like you have one cell line, sorry, and one virus, and just try to like go on and find out. So I was really excited about this project. So I'm still really excited about this project. So yeah, it was like understand if, of course, the viral insulin during the viral cycle is first produced, transcribed, and produced. Because if you have like something in your DNA, it doesn't mean that it's like transcribed and produced. So this is like very basic, but we have to understand it. And to do this, I did like just qPCR, like classical ones, as well as we don't know what is the fate of this insulin if they are secreted in the cells, outside of the cells, to have like autocrine, paracrine effects. Another really interesting experience like that I did like IF and mass spec to identify like the secreted proteins during viral infections. So this is like the first like step of the projects, and now we are like actively working on the in vivo project. We want like really to go on the organism. And what I didn't say is that all of these viruses belong to the Iridoviridae family, which are mainly fish viruses and reptile and a lot of like non-vertebrate viruses, organisms that can be infected by this virus. And so we are trying to find a good organism, and as we don't want to bring the real organism that can be infected by the fish, by the virus, which is the GIV. GIV virus is one of the viruses that I work with, and GIV is the short for grouper Iridoviridae. So we don't want to bring any grouper in our facility, or we could, but it's not as easy as it can be, of course, because they are quite large fishes. So the main model that I'm working on right now is the zebrafish. Makes sense because it's really convenient to work with. But the first things to do is to understand if they can be infected by our virus. It's of course not obvious that a fish virus can infect another fish. It's not like, so I'm really actively working on it. So I set up all the facility here to bring zebrafish and to perform infection on them. So this is like our main like actual big part of work on the in vivo part. And after, if we have like good results and if this fish can be infected, we will like start to understand first the tropism of these viruses, and if these VILPs, because I'm working, I'm at the beginning a molecular biologist, so my other part is to synthesize, to produce viruses that will be deleted for these VILPs. So in one hand, we will have the wild type virus, and on the other one, we will have the delta viral insulin viruses. And by playing with these two viruses, we will just understand maybe if there is a switch of tropism in the animal. As well, in vitro, we will do all the mechanistic and the like basic parameters that we know in virology, like replication curve and everything, to understand if these VILPs have a true impact on the viral cycle in vitro, but in vivo on the organs, on the different organs, and on the, as well, if we can go like really, if we look really far, we can like understand if it can impact the insulin and glucose homeostasis indirectly in the organism.
So zebrafish as a model system, I don't think we've talked to anyone that actually uses that. So can you talk a little bit about how do you infect zebrafish? And then maybe, if I'm not mistaken, one of the advantages to zebrafish is you can actually have, you can genetically not manipulate them in a way. So are you planning on doing that as well?
That's a good point. Yeah, the first point is how we infect the zebrafish, and it was like one of my first questions to answer, because how I was, thanks to my experience during my PhD, I was working on the Marek disease virus. It's a chicken virus, so I jumped from chicken to fish, which is kind of funny, but I have a good training on the Marek virus. And first idea was to do IP, intraperitoneal injection of the virus. And we have here some really good researchers that work specifically on the zebrafish, not on infectiology, but they give us really good clues and good help about this project. So yeah, one of the first experiments was to understand how we can infect it. So that's a really good question. So we try IP, I try water bath infection. People are doing this quite oftenly. So just basically put the virus in the water, like natural way of infection. And finally, we are ending up to infect the zebrafish by directly putting the virus in the gills and on the fish directly, to like really to avoid the dilution of the virus in the water, and to have, because it have a small impact on the potential infectivity. So we are working actually on this part. I did like several experiments, and the one that is relevant seems to be like directly on the gills of and also of the fish. And yeah, another point that I want to bring is, yeah, the tropism can also be impacted by the age of the organism. Of course, we know that some virus can infect children and not adults. It's exactly the same for fish. So we try different age of zebrafish, juvenile, larvae, embryo, and adult, and as well as different genotype. We have different genotype of zebrafish. And so that's the first part on the infection. And for the second part, I forget the question.
Oh, it's whether you're going to actually use the ability to genetically manipulate them.
Yeah, and we are actually working on, we already think about that, but to bring some immunocompromised fish to see if like there is any clearance by the zebrafish that will clear the virus out. So we try to bring some immunocompromised fish, so that are modified, to see if it's really like linked to the immune systems, or more really on the tropism, and that the receptor is not here. So we are not like actively working on some modified fish, because like we have so much things to do right now. But in the future, why not? Yeah, to like just suppress the interferon response in some fish and see if like this very specific virus can be like the curve and the growth of this virus can be modified. Yeah.
Cool. And is it only these invertebrates, as it were, that are invertebrate infecting DNA viruses that encode these viral insulins, or are there other sort of DNA viruses that also encode them?
No, they are all belonging to the Iridoviridae family. There is only 6 viruses that have this really interesting capability, but all of them like infect either fish. There is some really interesting data as well on frogs. But yeah, this is the viral part, and our lab is not a big lab, but we are like doing really like diverse experiments and like projects, which is really interesting. And another project that was just like published like really recently in PNAS, like our lab show as well that there is some bacteria that encode this insulin, and this bacteria could be involved in some onset of diabetes. So that's really interesting, and we just like our team just published a really interesting paper about that. So there is not only viruses, there is as well bacteria and other microorganisms that like encode this insulin. So there is viruses, but bacteria.
So it might be part of the microbiome, as it were.
Sure. Yeah. A lot of our lab, a lot of people in our lab are working on microbiome as well. Like the impact of the microbiome on the onset of disease is so like people are like getting are doing research so actively like you do on this part. It's so interesting. And as well for diabetes, there is like people that show that there is a higher prevalence of some subtype of enteroviruses in type 1 diabetes patients versus the lean. So yeah, it's really interesting to work here on this kind of interesting questions.
Right. Cool. And are you presenting some of this work at ASV this year or?
Sure. Yeah, I have a poster that I will present in Madison. It will be great to present here in person. So yeah, I will be happy to present this really interesting project to anyone that is interested in microbiome, but in virology. It's really like the crossing of lots of field here, and this is what I like in science. Even if I love virology at first, of course, but yeah, like doing biochemistry and microbiology in the same time, it's really interesting, and that's why I joined MRAS Lab at the first place. So yeah, I will present all our data that we get and the future. Sure.
Cool. And then I guess just to finish up, what has been, I guess you actually moved to Boston during the pandemic. What have the last 2 years been like for you? Not being sort of being in a different country, as it were, having to move, things like that.
Yeah, it was quite challenging last year to move, of course, due to COVID, and the US restriction was quite kind of strict. So we have to get like in national interest exception to come in the country. And it was challenging because at the end of our PhD, of my PhD, I have to just jump to the other country really shortly. But yeah, the main impact was before on my last year of PhD. It was really hard here because we locked down the country, and the lab was closed, and so we have to stop lots of really nice experiment on my last year. So this is a true impact here. But after, yeah, it was lots of paper to do, of course, to come to US. But after, when I was here, it was okay. And Boston is really a nice city, a bit cold when you're not used to, but it's really, yeah, really super interesting because we can have collaboration with a really wide range of people, zebrafish, people that are everywhere here, facility that are really innovative, and yeah, the environment here is really cool. So I'm really happy to be in Boston.
Great. Well, thanks so much for talking with us today, and we look forward to seeing your work at ASV.
Sure, Larissa. Happy to meet you there.
This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, and other podcast providers, or at lmtv.podbean.com.